I'm Joyce Robinson, and my group did the project about disrupting KPR function by studying how it would happen if we interfered with KPR dimerization, because changing the KPR structure can also change the function or inhibit it. So this should be useful in trying to make this study. We are studying KPR, which stands for ketopantoate reductase, and this is a key enzyme in the pathway to creating CoA. It starts with vitamin B5 and through steps converts ketopantoate into pantoate, and which is turned into pantothenic acid, which is vitamin B5, and is then turned into CoA. And CoA is the enzyme or the energy molecule of life. So if you do not have CoA, then you are not going to be able to survive, and you're not going to be able to do anything. So this is a key pathway in trying to inhibit the KPR, and this is going to be really important in medicine when we're trying to make medicines that will affect bacteria and not the human cells. So this brings us to why we are studying it. This could be a key pathway to medicine, like I just said, and it will actually affect human cells. But the difference is that bacterial cells will not be able to make vitamin B5 because of this pathway. And because it cannot make vitamin B5 when inhibiting KPR function, it is going to be difficult for them to make CoA. They're not going to be able to if they can't make KPR. But humans can eat vitamin B5, whereas bacteria make it through KPR. So if we can find a way to inhibit KPR function, then we will be able to have bacteria stop creating CoA because it can't make the vitamin B5 that's going to be turned into CoA. But humans will still be able to do so if they obtain vitamin B5 in their diet. This is going to be really important because if humans cannot make vitamin B5, then they have to eat it. And so the human is going to have to make sure that they are obtaining vitamin B5 in their diet so that their cells will also not stop producing CoA. This is going to be studied in many different ways through a series of several steps. The first is designing primers. Designing primers is going to be very important because primers are going to tell the polymerase where to cut and is going to take out this sequence to be able to create a mutation. This is relative to the E. coli plasmid, KPR, E. coli KPR plasmid. So it needs to be a sequence that is present in the E. coli KPR plasmid. These primers need to be designed with respect to each other, the forward and the reverse primers, as well as the restriction enzyme that is going to be used, and this will help us decide which restriction enzyme is going to be used. And this is going to help us do PCR because we're going to use these primers in the PCR reaction. PCR stands for polymerase chain reaction, and it's important because this is what allows us to create our mutant plasmid, hopefully. Hopefully it will work. And this is where the primers are going to be cut. They're going to be bound and cut and create a new sequence so that there will be the desired mutation. This will then be run on a gel electrophoresis to make sure that we have the desired results. And this will be determinant in if we're going to move on to our next step. If we were successful with PCR, then our gel should have 2 bands, one at the plasmid size of the DNA, of the plasmid, KPR plasmid DNA, and one at the site where the restriction enzymes were going to cut. Not the restriction enzymes, the polymerase. So then we're going to move on to transformation, where we're going to insert this mutant plasmid into the DNA or into the bacterial cells. So this is going to allow us to receive more plasmid with the mutation in it, so that we will be able to get a mutant KPR in the plasmid. And this is going to allow the bacteria to replicate and make more and more of this mutation, so that we will then be able to have the multiple copies of the mutation. And then the next step is going to be the plasmid purification, because all we want is the mutation. So all we want is the mutation in the plasmid. So we're going to do plasmid purification and get rid of all of the cell materials that are involved in this, like cell membranes, cell walls, genomic material, extra DNA, anything in the cell that is contained in a cell that is not part of the plasmid, because we only want the desired mutation. So this is going to help us to get the purified plasmid so that we can work on it without the effects of anything else in the cell. And then we're going to move on to restriction enzyme digestion. This is going to help us put an antibiotic resistance gene in the plasmid so that we can grow this in cells on a medium, LB agar, with an antibiotic in it. And it will allow us to see if our restriction enzyme is working and if we have the mutation that we would like to have. Because the restriction enzymes are going to bind to the desired location and incorporate an antibiotic resistance gene so that we can see if our restriction enzyme is working. And then after this happens, we are going to do another PCR reaction to make sure that the restriction enzyme is working. In this PCR reaction, we will then run a gel with the results from the PCR reaction. So we will be able to see if the restriction enzyme digestion was working. We should, on the gel, if we were successful, we should see a band, just 2 bands. One, that they need to be very close to each other. And one is going to be at the first base pair where it was cut. And the other one is going to be at the second. So if we are successful with this, then we need to make sure that we send this off to a lab that does DNA sequencing, because we are pretty sure at this point that we have the mutation that we need to be able to continue the experiment successfully. We do not have this methodology in our lab. We do not have the adequate resources to do DNA sequencing here, so we need to send the sample off to another lab where they can do DNA sequencing. And then when we receive the results back, we will see if we're successful by seeing if the mutation is present in this DNA sequenced plasmid. We don't want to continue on with this until we know for sure that we have the mutation, because we don't want to waste the resources, money, energy. We don't want to do any of that until we know for sure that we have the mutation of interest. And then once we receive the results back, we are going to do another transformation so that we can create for sure more of this mutated protein. We're going to incorporate it into a bacterial colony, into a bacteria, and let it grow on an LB agar plate containing an antibiotic. If you remember, we added the antibiotic resistance gene already, so it should grow. If it doesn't, then we have a problem and our restriction enzyme did not work. So we are going to grow it on an LB agar plate, and it should grow, and this should allow us to have more protein of interest, because the bacteria is replicating and making more of the mutated protein. So then, after we do this and are successful, we'll do another protein purification. This protein purification should be done in a similar way to the plasmid protein purification that we have already done. And then we will have the protein of interest and only that. So it'll be done by like column chromatography based on needs of the protein of interest. We will need to exploit the differences in the protein of interest against the other ones, against anything else that's in the cell, so that we can get rid of any cell material that is not the protein of interest, since we just want the protein of interest. And after this, we should be able to scale up our cells, because we have the protein of interest. We'll scale up our cells by allowing cells to grow with this, there's a bug, with this protein of interest incorporated into the medium. So then we should get more cells with more protein of interest. And keep in mind, our protein of interest is the KPR that's mutated, and it should not have a dimerization anymore. After we scale up the cells, we are going to be able to do an enzyme assay and test the differences in mutated KPR, the one that we have just created, and the original non-mutated KPR, and we'll be able to see the effects that the mutation has had on it. We'll be able to see if this mutation was successful in inhibiting the KPR through dimerization interference. These steps are important because for the reasons that I've already described, but each step is important in doing this experiment. So you can take a look just for extra reason we're doing this, and the more molecular way that we are doing this, is if you can see in these pictures, the top half of the picture is before the mutation, and the bottom half is after the mutation. You can see that we are trying to replace the glycine 194 with a mutation, because glycine 194 is involved in the dimerization. It's in purple, you can see here. In the top half, you can see that it is involved in dimerization and is making a bond with the orange piece. In the bottom half, after the mutation, this is what we expect to see. We expect that KPR inhibited will not have a dimeric interference engagement anymore, because we have intervened and disrupted this bond by changing glycine 194. If we change glycine 194, there should be no dimeric interface anymore, and this will change the structure of the KPR protein. We would expect to see less function of KPR in the conversion of ketopantoate to pantoate. And because of that, we will expect to see no pantothenic acid or pantoate or CoA, which is the main reason why we're doing this. We expect that CoA will not be made in the enzyme assay of the mutated KPR. So when we do the enzyme assay, we expect to see little function of KPR, and that means no CoA being made. This is important because this could be very beneficial when trying to make medicine, because if you can inhibit this in a bacteria or any cell really, then the bacteria are going to die, and the human cells don't have to. They might if you're not obtaining B5 in your diet, but if you know you are taking this drug, then that inhibits this process, and then you need to make sure that you are taking vitamin B5 or eating material that contains it. So this could be very important development in the field of medicine. This is a project that involves several steps, and checkpoints need to be made to make sure that it is being done efficiently, so that we do not waste time and resources on doing things that the previous step did not do right, and we do not have the thing that we need to do the next steps. So to summarize, we are trying to inhibit KPR function by inhibiting dimerization, by mutating glycine 194, and hopefully that will interfere with the KPR structure and the dimerization. If we inhibit this structure, then we can inhibit the function, because the protein structure is important in the function. So, and through a series of experimental steps, we can see what our mutation will be able to provide to this research. We expect that KPR will not be functioning after this mutation because of the structural changes that we have made to the KPR protein.